So my research is into how breast cancer spreads around the body and how we can stop it. Our research project is called Therapeutic Targeting of Dormant Breast Cancer Cells, the key to improving outcome for metastatic disease. And it's all about how we can kill off those cells that survive the first rounds of therapy and then cause the cancer to come back many years later. A dormant cancer cell is a cancer cell that isn't actually growing and being very active. It's just sitting there waiting for some signal to start to grow again. And this period can be really quite long. In breast cancer, it can be decades. And we don't understand exactly how the cancer cells survive this long without doing very much. And also, we don't understand enough about how they avoid being killed off by the therapies that we have today. We often call these dormant tumor cells chemoresistant in that they don't actually get killed by the chemotherapy drugs that we use to eliminate growing cells, cells that divide a lot. And that's the majority of breast cancer cells. Dormant cells aren't growing and dividing, so they are not sensitive to these chemotherapy drugs. And so we call them chemoresistant. All our research at the moment is on triple-negative breast cancer. So that means the type of cancer that doesn't express an estrogen receptor or progesterone receptor. We can't treat these with endocrine therapies that are so beneficial for the majority of breast cancer patients. And triple-negative disease is often much more aggressive than other forms of breast cancer. In our research, we try and use exactly the same chemotherapy drugs that most patients will be receiving during their treatment, because ultimately we want to understand how we can improve the outcome for patients, and it doesn't make sense to explore therapies that perhaps aren't ever going to make it into the clinical use. So we often use the very, very common drugs that most patients will be receiving. At the end of this project, we hope that we have developed some way of killing these dormant cells, or at least some way along the way to killing these dormant cells. It's not easy because they really don't do very much. And if you think of an analogy, I often say it's the difference between killing, for instance, a dandelion plant and trying to kill a dandelion seed. And you can see that the ways you would go about that would be very, very different. One relatively easy, one would hope, and the other one, killing the seed, is much, much harder. And that's what we're aiming to do with our research. If you want to know more about the research that we do in Sheffield, then please have a look at our website. The easiest thing is to put my name into Google, Professor Ingunn Holen, University of Sheffield, and you'll get a lot more information about the work that we do in my team.